Is Deepak Saini, who is a professor of developmental biology and genetics at the Indian Institute of Science. I think very interestingly, Deepak works in an initiative called Longevity India, where there are a number of institutes that report to each other on studies that they're doing with the elderly population and interventions. So a very interesting source of information. But today he's going to mostly talk to us about senescent or zombie cells and their ubiquitous role in the aging phenomenon. So please welcome Deepak.
Good afternoon, everyone. And first of all, thank you, Nadine and Martin, for giving us a platform, a platform to talk about what we're doing. Briefly, you mentioned that we have started an initiative called Longevity India. I'll talk about that a little later. But first, let's talk about some zombies amongst us or in our bodies. And we call these zombies the senescent cell. And we've heard a lot about them. And I'm going to tell you a little different story, a little hard on science. But I will tell you what makes this story unique is that we are trying to understand why certain therapies don't work in aged population. And also certain therapies don't work very well. So I'm going to talk to you about what things which are done in my group. So we've been looking at very fundamental questions in aging, which includes how DNA damage drives aging, how loss of protein folding or proteostasis changes during aging, how mitochondrial dysfunction creeps into aging, biomolecular oxidation, which will be the story for today, and how biomechanical alterations change during aging. Also, we do a lot of drug discovery, trying to look for new botanicals or new molecules which are in the traditional knowledge, how they can be used for anti-aging therapeutics. So we have slowly transitioned from cellular studies to multidisciplinary studies in human beings, and I'll talk about that right in the end. So for whom this talk is meant, who are the audience whom we think we should be listening to this talk? So people who work on senescence, age-related pathologies, immune and inflammation, hormone and reproductive aging, diagnostic, drug efficacy, anti-aging therapeutics. So you're wondering, how can I talk about all this in one single talk? And I'll take you through the journey, how we ended up unraveling something very interesting. I hope these videos play. There have a lot of videos in my presentation. I'm hoping they will play. So assume, since they're not playing, this is young cell, and these are old cells which we can grow them in the lab. And you can take any cell system in your lab. You can actually get them to behave like old system by giving them some insults. Now, insults are not really the insults what we say. We don't call them names. We just give them some DNA damaging agents. We give them oxidative stress. We give them radiation, chemotherapeutic drugs, and we end up getting this senescent cell in culture. And this phenomenon has been well established as one of the main drivers for systemic aging in our body. And what we started as a very curiosity, we wanted to look for a receptor, a GPCR, which is the most druggable molecule which we know of, that if there's a receptor which is uniquely present in senescent cell, and which we can use to target senescent cells and try possibly eliminate them from the body. And that's been the theme of most of the senolytics industry as of today. And when we looked at a lot of data set, we only found that one small receptor, CXCR4, and if there's clinicians, they know this receptor is also linked to very poor cancer prognosis. But this receptor was actually the same receptor which is present in aged cell at higher copy numbers. We were very curious that why a receptor which is used by aggressive cancer cells should be present on an aged cell. So we started asking question, what does this receptor do? And the movie is playing, so you can see how nicely the young cells divide. And these are the old cells. You can see they divide a few times and then just sit there static, not doing anything. The best part of these is senescent cells, you can keep them on this culture dish for weeks, months. They don't die. You actually don't need, you just need to feed them. They don't divide. They happily sit in the incubator. They are the worst nightmare for a PI and the best friends for students because they can grow these cells and go for a long vacation, come back, the cells are still alive to do the experiments. So when we look at this receptor in these cells, what we find that the receptor is upregulated in any kind of aged cell, whether it's aging being induced using a drug or a naturally aged cell, which is the last cell type, we see this receptor is being upregulated. I'm going to cut a very long story short here, but what we found that it was the presence of DNA damage which triggers this receptor copy number to go up, and which is this molecule called ATM kinase, which is the heart of aging or senescence program. that through presence of HIF1 alpha, which is stabilized by reactive oxygen species, is increasing this receptor, and this receptor actually increases inflammation. So the idea was very simple. The idea says that when the cells undergo damage, the receptor copy number goes up in these cells, which is this red cell, where you can see the cell is large and granular. And this copy number of the receptor goes up. And this high copy number receptor allows this cell to secrete more inflammatory cytokines surrounding this cell. The purpose of this more inflammatory cytokine is to recruit immune cell to clear this damaged or senescent cell, which makes a lot of sense because senescent cells in our body are generated on an everyday basis, and they need to be cleared up. And this is a very simple mechanism by which immune cell can home onto the damaged cell in your body when the billions and trillions of cells are sitting together. So this was something which was very, very interesting, and we said maybe this is a receptor we can target to prevent inflammation from occurring in senescent cell. But the problem was if you stop that, then senescent cells are not going to clear by your body and they'll accumulate. So we said, okay, we have hit a bummer, and this is not going to be something which we can explore any further. But what happened, there was a serendipitous thing. We actually, one of my students left the senescent cell for a little long time. And when we did this experiment, and when the cells were senescent, just entering senescence, we found the inflammation is going up, which is what we want to see because we want immune cells to home. But when we left the senescence for a very long time, actually when we activate the receptor, the inflammation was not going up, which is very peculiar because we thought if the same receptor is actually causing inflammation activation, why should the receptor activation cease, even though the receptor copy number is very high in deeply senescent cell? Now this is where we started to understand this receptor, which is ubiquitously present. We cannot knock down this receptor. We cannot block this receptor. But this receptor is there in the aged cell, and how it is not behaving the way it is supposed to behave. It was very curious for us because it also showed or gave us a hint that many receptors, which are the target for most pharmaceutical molecules, may not be working the same way in the aged condition. So this is where we started investigating, and we found something very fascinating happening. So I'm going to show these movies again. I hope they play. I hope so. Okay. Yeah, you can see that. This is what happens in a young cell. You can see these fireworks. These fireworks are basically calcium oscillations. So all cells in our body show calcium oscillations, and they show calcium release, calcium absorption, and we can actually track them by giving a dye, which binds to calcium and starts to glow. So every time the calcium comes out, cells glow, and they come back and they start glowing it again. So it follows a very beautiful oscillatory pattern, and which you can see that. And this is what happens in young cell. But when you take the old cell, the oscillations disappear. Actually, you can see there's only one peak or there's a flat line. So that means old cells have changed their behavior in terms of one readout, which we had was calcium release or calcium oscillations. That made us to ask the question, why was the receptor behaving very differently? Then we started looking at the membrane of these cells. We found the membrane permeability was different, the membrane fluidity was different, and the stiffness of membrane was different. So everything was telling us there is something wrong with the lipid of the cell or the cholesterol content. So all evidences pointed towards cholesterol depletion from the aged membrane, which is very strange because you say cholesterol goes up during aging. So we started asking the question by looking at the content of cholesterol in the aged cell. Now these experiments are done in a whole lot of different types of cells, but what we found that in the aged cell, the cholesterol was interestingly undergoing oxidation. Now, a lot of clinicians, and I hope here, understand that oxysterol is actually the major culprit of age-associated pathologies. And we have wanted to see if this oxysterol, which is causing all the age-related problem, is the one which actually changes the behavior of the receptor on the membrane. So cholesterol can oxidize into oxysterol at multiple positions, and oxysterol is different in terms of hydrophobicity. You can have oxidation on the tail or the head, and basically when there's more oxysterol, the membrane actually undergoes disruption. And when membrane is disrupted, the aged cell actually starts behaving differently. So what we did, we started quantifying cholesterol content in the young and old cell by using mass spectrometry. And what we found that in the aged cell, cholesterol levels were not changing, but it was actually the oxysterol, the oxidized form of cholesterol, which was increasing in almost all the different oxysterols which we measured. We measured them across different cell types, different conditions, and what we find that in aging, oxysterol is actually, which is going up. And so it's not the cholesterol which we should be measuring during aging, it's at the level of oxysterol which needs to be recorded. So what does the oxysterol does? So what we found interestingly that if you actually look at the ratios, because oxysterol increase in your bloodstream is telling your body there's too much cholesterol, stop making cholesterol. So the cell actually stops synthesizing their own cholesterol, and the cell starts behaving as if there's a lot of cholesterol in the body, but there is not. So cholesterol turns out to be a natural antioxidant in your body, which needs to be there to actually prevent damage by reactive oxygen species to the cells. So this figure actually shows the cholesterol is low and oxysterol is higher. But where am I going with this story? So what we actually found, that if you take young cells and you actually throw oxysterols on them, just oxysterol, no damage, no distress, no oxidative stress, you actually can cause aging in the cells. And this can be seen by multiple oxysterols, that you can see the calcium oscillations are present with some oxys give you a flat line, and some oxys actually knock out the activity completely. So just accumulation of oxysterol, which is actually an indication of poor liver health in your body, leads to senescence. So it could be all burning down to what is your liver or metabolic health. Now, this is just, again, a video to show what happens in normal cells and what happens in oxysterol-enriched cells, that they actually don't show any calcium oscillations. So we went back on the drawing board to ask, is this information trying to tell us something? So we went back to the CXCR4 receptor, which is what we studied. We did a lot of molecular dynamic simulations. And the box region, which you can see here, actually shows you where the receptor, these are the various amino acid positions to which the receptor binds to G protein, which is responsible for their activity. And we found that presence of oxysterol actually prevents G protein coupling. Now, this got a very important implication. Most of the drugs which we consume pharmacologically, they actually target GPCRs, whether it's antihistamines, antiadrenergics, muscarinic acid, nicotinic acid. They're all targeting GPCRs because we want the GPCRs to be inhibited or activated. And most of the drugs are actually consumed by elderly, even though in the trials we actually eliminate people age 55 and above because they give very heterogeneous clinical readouts. So we do not want them to be included in the trial. But in real case, it's the people which are age of 55 and above who are consuming these drugs. Based on this information, what we realize that most receptors actually don't behave the way they should behave in old cells. And if you're trying to give that drug to an old cell, it just might flip their activity or behavior because of the simple change in the cholesterol to oxysterol ratios inside the cell. So that actually beckons for us to understand the pharmacology of new GPCR agonists or all the existing GPR agonists, that if they're actually working the same way in elderly, and if there are a lot of complications which we see in elderly are coming because of these drug molecules which are being used in elderly, assuming that's the way they work in elderly. Now, assuming what we were right about oxidation of cholesterol, we actually did a very simple experiment. We prevented oxidation of cholesterol, and we see if we prevent oxidation of cholesterol using antioxidants like quercetin here. We also tried other molecules. Can we restore the oscillations back? And yes, we could restore the oscillations back. That means there is a merit in having antioxidant-rich diet, which actually affects how this molecule oxidation occurs in a real scenario. So this is the big picture which we have actually identified, that the cholesterol oxidation into oxysterol. One of the speakers previously talked about cholesterol role in aging. We actually see how oxysterol in aging affects how the receptors regulate the inflammatory responses based on a fundamental point of view, but it also shows you how receptor oxidation or the cholesterol oxidation changes the receptor conformation, thereby changing the way the drug molecules actually work on these receptor molecules. So this becomes us to tell us that the real bad cholesterol is actually the oxysterol, and oxysterol is linked to lysosomal dysfunction, linked to liver changes. Brain oxysterol is linked to dementia. Impaired cholesterol is linked to macular degeneration, also for liver cirrhosis. So I think we need to change our way of measuring cholesterol and start looking at oxysterol molecule. Now, we also did something more. We actually showed that just addition of oxysterol is enough to cause metabolic distress in the cell and also change the mitochondrial activity in the cell. These are all normal young cells, and you just add oxysterol, they actually start behaving like a super-aged cell in all the parameters which we know about aging. And this brings us to what we are actually doing in our cohort in India. We have a study, ongoing study called the Bharat study. I'll tell you about what exactly it stands for. And we also found in the aged population here, especially in this study, we find the cholesterol to be elevated, but oxysterol was actually much more elevated. So if we keep this information in mind, that it's a ratio of cholesterol to oxysterol, which dictates the physiology, then we really need to look at new diagnostics, new drug delivery, discovery, how the receptors, which are conventionally or currently being used as targets for all the various pathologies, how does their behavior change? So keeping this pathway in mind, what we have started doing is we started doing a drug discovery, screening for molecules, and what we found that we have some molecules in the cholesterol metabolism pathway which can work as senolytics, and they are, when pegged against the conventionally well-utilized senolytics like ABT, they tend to behave better. So this molecule is currently under investigation. We have done mouse model studies. I'm not showing you the data, but what we found that in older mouse, if we actually block the cholesterol oxidation process and also replace the cholesterol level to the normal level, we actually see better neurocognitive function, memory restoration, and they tend to do better in maze test. That also raises a very interesting point. Cholesterol is actually the precursor molecule for hormones. But if cholesterol goes oxidized, it becomes oxidized oxysterol, it cannot serve as a precursor for the reproductive hormones. That raises a very interesting question that if you are actually getting old and oxysterol levels in your blood are increasing, you're actually going to have lower levels of hormones in your body, which means estrogen, androgen, and it's indeed true in the older, as people get old, the level of these hormones fall. We have not measured, I don't think anybody has measured the ratio of the hormones to oxysterol, but that's an ongoing exercise which you would like to do and understand if oxidation of cholesterol is an underlying cause of hormonal basis of aging, which we see in elderly. So these are the key findings from this study, that oxidative stress drives cholesterol oxidation, which alters membrane dynamics, which changes how the receptors, which are the key drug targets, alter their behavior, and how oxysterol needs to be measured as a factor of the total cholesterol in the system, because drug pharmacology must be evaluated in ratio of cholesterol to oxysterol. And oxysterol might be the next marker which we can use to understand physiological pathology in the aged. Now I'll move to what we are doing in India in terms of longevity space. So while we were doing this lab-based research, we realized that a lot of my team members were doing a lot of interesting aging studies in isolation. So we said, why don't we start putting our forces together and we actually start asking questions which is more relevant to human and more relevant to India. We've heard a lot of speakers talk since morning about clocks, various biomarkers of aging, healthy aging, but clearly we also understood that exposomes are the one which drive the signatures of aging, environmental factors. And environmental factors are different for different population. In India, the exposome is our gut and our lung, which is very, very different because our air quality and food we eat is very different, and that might be the case for every place. So what we did, we started looking at Indian population from the more granular lens. An idea was that all the experts which are there in our study group will study Indian population, and we start a study which is called the Bharat study, which is biomarkers for healthy aging, resilience, adversity, and transition. It's an acronym for Bharat. What we do, we look at healthy people. The healthy people don't come to hospital. Again, in a country like India, no matter how many subjective studies, always less. So we reach out to various rural settings, we reach out to voluntary recruitment, and we collect data from each individual. And our data for healthy people, we cannot subject them to routine MRI, X-ray, because ethically it's not allowed for us. So what we do is you collect bloods, stool, hair samples, urine, and cheek swab, and we are using that data and samples. We collect almost 2 million data points per person. And the idea is to build that data set to understand aging trajectories in India, which could be resolved on the basis of geography, diet, socioeconomic background, rural-urban divide, genders, and we get the new baseline values for health for India. And the idea is using this information, we can also do future biomarker discovery. There's another thing which we do under this initiative is we make 3D organ systems for aging, which are multicellular organ system. Like, for example, we are making a lung-on-chip model, where we give breathing ability to the 3D printed lung, liver, blood vessels, and use this model system to actually mimic at multi-omics level the actual human data, and use these model systems for therapeutic drug discovery and to look at efficacy of existing drug molecules. What we have found that using these, some of the platforms, especially lung, liver, and skin, that the top 20 selling drugs, actually most of them don't really have any impact on the pathology of aging cells. So we have aging cell model systems, which we use in this copycat models and understand how they actually undergo aging. Then we have something which we, on a lighter side, called grandma therapeutics. A country like India is very rich in terms of its traditional knowledge. We have a lot of botanicals. We have a lot of therapeutics, which has traditionally been used in India to counteract various age-related changes in your body. And these recipes exist in small clusters, small regions of India, where this information can be extracted and documented. We also call it that traditional knowledge. In some forms, it's called Ayurveda. Again, it doesn't have a lot of evidence whether it works or not. So what we're doing, we use this traditional knowledge and we validate them at multi-scale. We actually characterize the biomolecules. We extract the biomolecules, test them in our model systems to see if they have any positive effect or they work. And then we also use them in our animal and also test them in human trials to see, because they're already being consumed by the population, to see if those people who are using these molecules actually show better physiological outcomes or not. Then we also have a very major outreach program. In India, aging is a state of body which we call as denial. So we deny that we are getting old because a lot of Indian, almost 60% of Indian are in the rural setting. In the rural setting, we actually have a very interesting conundrum that the healthcare access, availability, and availability in time is actually very limited. So a lot of elderly population don't want to know if there's something wrong with their body. Because if there's something wrong, they actually become an economic burden to the societal system. So people just don't want to know if anything is wrong. And so what we do is in our outreach program, we actually have ambassadors in various education institutes in the country where people reach out and educate elderly about healthcare needs and how lifespan and health span is slightly different. We actually teach them about how you can improve the quality of your life if we actually do early intervention, early diagnosis. We also work with a lot of industry, a lot of policymakers, and I'll talk about what do we do there. We have active podcasts, we release newsletters, a lot of meetings and collaborations as run pan-India to make sure that the longevity, which is the health span extension paradigm, is reaching to all the people. And this is the detail of the Bharat study. We collect a lot of functional data, cognitive data, metabolic data, metabolomics, proteomics, lipidomics from the serum, urine and stool, gut biome analysis, biophysical changes in immune profile. The aim is to do at least for 10,000 people in India, 2 million data points. I think that will be sufficient meat or material for the AI guys to do and train some model which are very India-specific. And using this information, we would like to develop organ-specific clocks for aging, where we can understand what changes occur in the body of Indians, where we can use future discovery. So this is Longevity India's broad umbrella. We have a very big team of almost 30 investigators who do deep science. We have almost 20 clinical hospitals as a part of this initiative, which do community medicine. We work with almost 12 companies who actually develop technologies, low-cost diagnostic, which is field deployable. Also the findings, some of the speakers in the morning said, let's see, I find markers, what do I do about them? So we work with companies and try to develop those markers as point-of-care devices or bring the cost down so they can be deployed in field. And we work with policymakers. For example, last year, we actually got lifestyle as a medicine program registered as an MD program. So now clinicians in India can register for an MD in lifestyle medicine, because lifestyle is at the heart of aging-related problems. So that is the part which we do. So there's a lot of interesting initiatives which we have started in the last 1 year. And these are our team. It's a pretty large team, almost 25 different investigators from deep sciences there. And I'll stop here, and I'll be open to any questions. Thank you.
Thank you, Deepak. That was great. I love the comment about being in denial. I think Indian and Irish people need to swap around a little bit because
Oh, is it?
we spend our whole life talking about how old we get and how we
Yes,
feel old and how we...
I think in India, it's like, as long as you deny you're old, you're good.
Okay, okay, it's a different approach. So there's a few questions coming in. If we can just tackle one or two. What do we know about today where senescent cells accumulate first in the human body? And does this vary by lifestyle, genetics, or environment?
Okay, what do we know today about where senescent cell? That's a fantastic question. You know, if you're, I don't know how many of you have been to India. Some of you have roots to India, I'm sure. But if you're in Delhi, I'm absolutely confident the senescent cell starts in lung. Because that's environment. Exposome is that bad, that so much of particulate matter, it'll start in lung. But if you're in some other place, if it depends on what kind of lifestyle you have, certain type of foods will trigger senescence in gut. Certain type of food and nutritional habits will change senescence in liver. So it depends on what is the environment doing to your body. So it's no one single site which will start the senescence process or aging process. Actually, one of the main themes of systemic aging in human is to find that weak link in your body, which is aging at a much faster rate than the rest of your body. So if you find that organ, whose clock is at a faster rate, you can actually fix that organ, slow the systemic aging. I think that would be the best way to take it forward.
So just to follow up on that, because this is something a lot of the biological clocks that were discussed earlier are looking for a unitary biological age for someone, whereas I have seen some work that would look at a multi-organ.
Exactly. So we need to have an organ-specific aging clock, where each organ is ticking at a different rate, and find the weakest one, the one which is ticking at the fastest rate, and fix that. I think your systemic aging will slow down.
And can there be, but can there be a substantial difference between different organs within the same individual and how they age?
Yes, I would.
So my heart is older than my...
Absolutely, absolutely. It'll be surprising if it's not so.
Okay, geez, very good. Do you want to take the top question real quick?
Yes, so the next one question is, do you see a toxicity difference between enzymatically produced oxy and non-enzymatic oxy? Yes, the answer is yes. There is a difference. So like the tail oxidized oxys tend to be more deleterious than the head oxidized cholesterol. So there is a difference. And we still do not know how much of that comes enzymatically, how much of it that comes non-enzymatically.
Amazing.
And there's one more question. Does a patient on chemo drugs have more side effect and adverse effect because of oxidation of cholesterol, as well as it a link? I would guess so. Again, something which we have not looked into, but I would guess it would be one of the features that people who undergo chemo actually undergoes more oxidation of cholesterol.
Brilliant.
Yeah, you just have to check it. We have not really investigated oxidized cholesterol as a mainstream molecule, and it needs to be done.
Amazing. Deepak, thank you very much.
Thank you.
Thank you.